In this video, we are going to take a look at some of the factors that affect enzyme activity. So we're going to take a closer look at enzyme concentration, substrate concentration, and remember the substrate is also known as the reactant. We will also take a look at how other factors like temperature, pH, and salinity may affect enzyme activity. And when we're talking about affecting enzyme activity, we're talking about the fact that enzymes work best in some situations as compared to other situations. So based on these factors, sometimes the reaction rate is increased and other times it is decreased. So let's start by talking about enzyme concentration. And concentration just refers to how much enzyme is present. Generally speaking, as the enzyme concentration increases, the reaction rate will also increase. And we see this in the first part of the graph here as the enzyme concentration goes up. So for example, if we start with 0 enzymes, and then let's say we add 1 enzyme, 2, 3, 4, etc., we'll see that as the enzyme concentration increases, the reaction rate is also increasing. But you'll notice that it reaches a point where that reaction starts to level off. So it does get to a point where adding more enzymes does not speed up the rate of the reaction. And we'll go into that a little bit more detail on the next page. So let's take a look at some numbers to kind of help illustrate how the enzyme concentration affects the reaction rate of an enzyme catalyzed reaction. So let's put together a little chart to help us keep track of our data. So first of all, let's take a look at the number of substrate molecules present. In the next column, we'll take a look at the number of enzyme molecules present. In the next column, we'll take a look at how long it takes for a reaction to occur to break down all of the substrate molecules. So we'll just call that the overall reaction time. And then in our last category, we'll take a look at the reaction rate. So if the overall reaction time, we'll use seconds. So we'll put a unit in there for seconds. So for the reaction rate, we're looking at a ratio of how many reactions can be catalyzed per second. So we can even maybe kind of put down, give it a unit of substrates broken down per second. Now, for the sake of this demonstration, we're going to keep the number of substrate molecules the same. So we're not going to change the number of substrate molecules present. We're only going to change the number of enzyme molecules present. So let's say we start out with 6 substrate molecules. And let's say we only have 1 enzyme molecule. Now, let's assume that an enzyme takes 1 second to break down the substrate. So like I said, that's an assumption we're making. So we'll put that up here. So we'll assume that 1 enzyme can break down 1 substrate in 1 second. So what this assumption means is that if we have 1 enzyme molecule and we have 6 substrate molecules, it would take 6 seconds in order for that 1 enzyme molecule to break down all of those substrate molecules. So if we were to look at the rate, that would give us 6 substrate molecules broken down in 6 seconds, which gives us a rate of 1 substrate per second. Now let's see what happens if we add another enzyme molecule. So let's keep the number of substrate molecules the same. So we still have 6 substrate molecules. But now let's say we have 2 enzyme molecules. Now each of those enzymes can be working at the same time. So while one enzyme is catalyzing one reaction, the other enzyme can be catalyzing the reaction with another substrate. So the overall reaction time would take 3 seconds. So in doing so, we would end up with 6 substrate molecules broken down in 3 seconds, which gives us a rate of 2 substrate molecules per second. If we were to increase the number of enzyme molecules again, so once again, we'll stick with 6 substrate molecules, but now let's go ahead and jump and say we have 6 enzyme molecules. Now, each one of those enzyme molecules can break down one substrate at the same time. So it would only take 1 second in order for all the substrate to be broken down. So that would give us a reaction rate of 6 substrates broken down in 1 second. So that gives us a rate of 6 substrates per second. So we can see here that our rate is increasing because we started out with 1 substrate per second, and now we've gone up to 2 and now up to 6. Remember, a rate is kind of like a speed. It's how quickly something happens in a given amount of time. So by increasing the number of enzyme molecules present, meaning we're increasing the concentration of the enzyme molecules, we're also increasing how quickly that reaction is happening. So we're increasing the rate at which the substrate molecules are being broken down. Now let's keep going. So let's say that we have our 6 substrate molecules, and now let's double our enzyme molecules. So now let's say we have 12 enzyme molecules. The overall reaction time is still going to be 1. And you might be wondering why that's the case. And the case is because the substrate is now the limiting factor. And we can actually look at this, this number 12 as almost kind of being like 6 plus 6, where 6 of those enzymes are catalyzing the reaction of 6 of those substrate molecules. The other 6 enzymes are just kind of hanging out with nothing to do because there aren't any substrates in order for them to work upon. So what happens is we end up with 6 substrates being broken down in 1 second for a rate of 6 substrates per second. So you'll notice that we have not changed from the previous example. So when we had 6 enzyme molecules, our rate was 6 substrates per second being broken down. Now that we have 12 enzyme molecules, we're still at 6 substrate molecules per second being broken down. So in the first 3 examples, our reaction rate increased, but we got to a point where the substrate molecules became a limiting factor and that reaction rate leveled off. We could go one step further. And let's say we still have our 6 substrate molecules, but now we have 24 enzyme molecules. Now, once again, we could kind of break this down and say that we have 6 enzyme molecules that are going to be breaking down these 6 substrate molecules. But we have 18 enzyme molecules that are just sitting there with nothing to do because there is not enough substrate available for it to catalyze a reaction with. So they're adding these extra enzymes are not increasing the reaction rate because the enzymes don't have anything to react with. And so we end up with an overall reaction time of 1 second. And so once again, we end up with 6 substrates being broken down in 1 second for a reaction rate of 6 substrates per second. And so once again, our reaction rate has leveled off. It increased at first, but then it leveled off. So now let's take a look at the data from the previous page and see what it looks like if we were to put it on a graph. So I transferred over the information looking at just the enzyme concentration and the reaction rate since everything else stayed the same. So if we take a look at our graph over here, you'll see that we have enzyme concentration on the x-axis and reaction rate in substrates per second on the y-axis. Now on the x-axis, we're going to make each square equal to 1 unit of enzyme concentration. But we have 24 squares that we would have to label, so we're just going to label every other one. On the y-axis, our data ranges from 1 substrate per second to 6 substrates per second. So we'll make 2 boxes equal to 1 unit on the y-axis. Now we can start out by graphing our data by putting a data point at the origin, because we can assume that if there are no enzymes present, there will not be a reaction rate. Then to continue with the graphing, an enzyme concentration of 1 gives us a reaction rate of 1. An enzyme concentration of 2 gives us a reaction rate of 2. An enzyme concentration of 6 gives us a reaction rate of 6. And then an enzyme concentration of 12 also gives us a reaction rate of 6. And an enzyme concentration of 24 also gives us a reaction rate of 6. So as we connect our data points, we'll see this trend with the reaction rate increasing as the enzyme concentration increases until it levels out. Now remember, it levels out because we have essentially run out of substrate to break down. And so adding more enzyme does not speed up the reaction rate anymore because all of the substrate has already been reacted upon. Now let's take a look at the effect of substrate concentration on reaction rate. And remember that the substrate is also called the reactant. So this is the molecule that is reacting with the enzymes. So as the substrate concentration increases, the reaction rate also increases to a point, and then it starts to level off. You'll notice that this graph looks very similar to the graph comparing enzyme concentration to reaction rate. As we add more substrates initially, there are more collisions between enzymes and the substrate, and the reaction rate increases. But eventually, all the enzymes are saturated, meaning that all of them are busy breaking down substrates. So adding additional substrate will not speed up the reaction rate because all the enzymes are busy. So let's take a look at some hypothetical numbers to better understand why substrate concentration affects reaction rate the way it does. So first we're going to start by looking at the enzyme concentration. And we're going to keep this consistent through our experiment. So let's say that we have 6 enzymes in our solution. So we have a concentration of 6 enzymes per solution. Next, we are going to look at the substrate concentration. And for the substrate concentration, remember this is the thing that we're kind of testing. So we're going to be increasing the substrate concentration, and we'll use numbers similar to what we used in the previous example where we looked at enzyme concentration changing. So we'll look at 1 substrate molecule, 2, 6, 12, and 24. Next, we will take a look at how quickly the reaction occurs. So we'll look at total reaction time in seconds. And then we'll look at reaction rate, which would be the number of substrates or the number of reactions per second. So first looking at 1 substrate. So if we have 1 substrate present and we have 6 enzymes present, and once again, we're going to make the assumption that it takes an enzyme 1 second to break down a substrate. So if we only have one substrate molecule present and we have 6 enzymes present, really we have one enzyme molecule that's going to break down the substrate, and then we're going to have 5 that are just going to be hanging out because there is not enough substrate for those additional 5 enzymes to work on anything. So the total reaction time is going to take 1 second. And the reaction rate is going to be one substrate. So one substrate molecule being broken down in 1 second. So that gives us a reaction rate of 1 substrate per second. Now let's say we have 6 enzymes, but we only have 2 substrate molecules. Once again, essentially 2 of those enzyme molecules are going to be breaking down the substrates in 1 second, and then the additional 4 enzyme molecules are just hanging out because they don't have anything to work on because the 2 substrates will already be broken down. So we will end up with a total reaction time, once again, of 1 second. But this time we have 2 substrates that were broken down in that 1 second. So we're going to end up with a reaction rate of 2 substrates per second. If we have 6 enzymes and 6 substrate molecules, then each of those enzymes will be working on 1 substrate molecule at a time. We'll have a total reaction time of 1. But we have 6 substrates being broken down in that 1 second. So now we have a reaction rate of 6. Now, let's say we increase the substrate concentration. So now we have 12 substrates. Well, 6 of those enzymes can break down 6 of those substrates in 1 second. And then remember that enzymes are reusable. So those same 6 enzymes can then break down the remaining 6 substrates in another second. So it would take 2 seconds for those 6 enzymes to break down those 12 substrate molecules. So what we would end up with is 12 substrates being broken down in 2 seconds. And we can simplify that just like a fraction. So 12 divided by 2 gives us a reaction rate of 6. Once again, if we increase that substrate concentration to 24, now we essentially have 6 times 4, right? So we have 6 substrates that are going to be broken down in 1 second, plus 6 more substrates being broken down in a second, plus 6 more, because 24 divided by 6 gives us 4. So essentially what we end up with is that it takes 4 seconds for these 6 enzymes to break down these 24 substrate molecules. So what we end up with are 24 substrate molecules broken down in 4 seconds. And that gives us a reaction rate of 6 reactions per second, or 6 substrates being broken down per second. Now, you'll notice that these numbers look very similar to what we saw when we looked at the effect of enzyme concentration on reaction rate. So I'm not going to regraph this, but once again, we're going to get a graph that looks very similar to an increase initially, but then it levels off.
Enzyme activity is also affected by temperature, and most enzymes have an optimum temperature at which they work the best. And so you can see this graph here that this is showing a temperature of 37 degrees, and this would be 37 degrees Celsius. And 37 degrees Celsius is normal body temperature. Or I should say normal human body temperature. And so this particular graph is showing you that this enzyme works the most efficient, or works the best, at that 37 degrees Celsius. Now, some enzymes are optimized to work better at different temperatures. So there's some enzymes that work really well in really hot environments, and other enzymes that work really well in really cold environments. And so the graph would look very similar in that it would have this kind of curved shape to it, but the curve would maybe be in a different part of the graph, depending on where that enzyme is optimized to work. So typically what happens is if the temperature is too high, there's actually too much agitation. So molecules all have their own kinetic energy. So the enzymes and the substrates are all just kind of bouncing around, and they're bouncing into each other. And as temperature increases, those molecules start moving around faster. And if they move around too fast, then when they bump into each other, instead of forming a bond, they actually kind of bump away from each other, and that bond is not formed, and the enzyme can't work on the substrate. Sometimes if temperature is too high, the enzyme might actually become denatured. So remember that enzymes are proteins. And proteins have a three-dimensional shape. And that three-dimensional shape is really important. So we've talked a little bit about how enzymes and their substrates have this kind of lock and key or puzzle piece model. And that's because the enzyme has to match the shape of the substrate in order for it to work properly. So if you add too much temperature and it causes the three-dimensional shape of the protein to unravel, it's kind of like melting the key. So if you were to take the key to your door and melt it down, it's no longer going to fit in the keyhole and it's not going to unlock your door. If the temperature is too low, we kind of have the opposite problem in that the molecules actually slow down, so their kinetic energy slows down. And as these molecules are moving slower, there are not enough collisions or interactions between the enzyme and the substrate in order for the chemical reactions to be catalyzed as efficiently as possible.
Enzyme activity is also affected by pH, which is a measure of acidity. So if the pH is not ideal, so if it's not the optimum range for the enzyme is just not going to work very well. So they might lose their 3D shape. So once again, remember that shape is really important. If the shape of the enzyme changes, it can no longer do its job. And we call that being denatured. And so if the pH is not in the optimum range, then the active site of the enzyme and the substrate are not going to be able to meet up properly. So in humans, the enzymes that are in our blood typically work best in a pH environment of 6 to 8. And that's because the human blood pH is normally around 7.4. And many of our body fluids are also around that slightly basic kind of 7.4 pH. However, we do have different components in our body, or different compartments, if you will, that have different pHs. For example, we know that the stomach is really acidic, and that acidity is actually really important for enzyme activity because there's an enzyme called pepsin, and that pepsin only works in a really acidic environment. So without the acidity in the stomach, the pepsin cannot break down proteins in the stomach. Now, interestingly, trypsin is an enzyme found in the small intestine of humans, and it has an ideal pH of 8. And it's interesting because as the pepsin from the stomach moves into the trypsin, the pepsin actually becomes denatured, and then the trypsin actually becomes activated. So our body actually controls which enzymes are active by changing the pH environment of different parts of our body. Now, we didn't really talk about salinity, but salinity is also something that can affect enzyme activity. And by salinity, we mean basically saltiness. So, how much salt is present or how much ions are present. And even though we didn't talk about salinity specifically, it would have a similar curve as what we saw with enzyme activity and pH and temperature, where there would be an optimum range at which that enzyme would work best. So enzymes that work in the Dead Sea, for example, would work really well in a really saline or a really salty environment. Enzymes that work in the ocean work well in a salty environment. But enzymes that work well in freshwater would be denatured in the salt water of the ocean and vice versa. Now let's talk a little bit more about this word denature. So remember that proteins have a three-dimensional shape. Now remember that enzymes are mostly made up of proteins. There are some exceptions to that, but most enzymes are types of proteins. Now, that three-dimensional shape really makes a difference. So, if the shape changes, the enzyme is no longer able to match the substrate like puzzle pieces or like a lock and key, and the enzyme can no longer catalyze the reaction. So you can see this picture down here of a raw egg and a cooked egg. And this is a very clear example of what proteins are like when they are denatured. So an egg is made up of a lot of protein in both the yolk and the white. And when you apply heat to that egg, it cooks it. And you've probably noticed that there are a lot of properties to that egg that change. So the color changes, the texture changes, the taste changes. And all these changes are due to that protein being denatured by heat. Another example would be something like ceviche. If you've ever had ceviche, it's where they take usually like fish or shrimp or something along with some vegetables, and they soak it in an acid, usually lemon or lime juice. And the acidity denatures the proteins in the fish and kind of cooks it in a way. Other examples include things like pickling. So if you take cucumbers and you put them in a solution that's really salty and really acidic, it denatures the cucumber and it forms pickles. And so these are just all examples of how these proteins are changing when they're put into an environment that's not ideal. And in the case of, cooked eggs and ceviche and pickles, these are all yummy things that we eat. But if these were the enzymes in your body and they all of a sudden experienced heat that they weren't supposed to experience, or pH or salinity that was outside of their optimal range, they could stop working. And basically, when your enzymes stop working, you stop working. And so if your enzymes become denatured, then that can kill you. So this is one reason why homeostasis in living organisms is so important. So homeostasis ensures that those optimal ranges are maintained so that your enzymes do not become denatured. Now, one thing I want to point out, and this is kind of a pet peeve of mine, but enzymes cannot die. So you cannot kill and they cannot die. And that's because enzymes are not alive. Enzymes are just molecules, they're proteins. Proteins are not alive, they don't have all of the characteristics of living things. So you can't kill an enzyme, and an enzyme can't die. You can denature it though. So just that's a little bit of a pet peeve of mine when people use that terminology, because you can't kill something that's not alive to begin with.